Hello everyone and welcome back to my channel Be Yourself. This is Dr. Rajni Sharma and today I will be discussing about the hypersensitivity. This is in the continuation with the series of immunology. So today let's start with the hypersensitivity, and this is a very common topic where a maximum population is being affected of hypersensitivity. Some are having very severe symptoms or some are having very minor of this. So let's start with this, what exactly it is. But before going forward, I will like to say that this topic is also very important for a competitive examination, as I have noted some of the questions from the previous past year questions from PhD entrance examination. Now, I have included only two of the entrance examination. One is the CSIR JRF NET and another is the ISC Bangalore entrance examination. But there are others examination also where this hypersensitivity is a very major topic. So you can pause this video and go through this all these questions which is been written here. I will better discuss with this topic, and then at the end I will come to this to all these questions so that you can answer all these questions by yourself. So basically, from the hypersensitivity, not very difficult questions or conceptual questions can be asked. So if you have a grip on this topic, then it will be a scoring topic for you all, because direct questions are actually asked from this. So let's start with the topic, and at the end we will come to the questions. So first I will start with the history of this, that how it was concluded that such types of reaction was there. So this credit goes to the king of Morocco, who observed that the people who were taking bath in the Mediterranean Sea, they were usually affected of some types of hypersensitivity, and that was due to the some toxins released from the jellyfish. So what the king did, he contacted two scientists who were the Portier and Charles Richet, and when they analyze this, what they did, they find that this is all because of this jellyfish or some toxins released from jellyfish, that the persons who are bathing in the Mediterranean Sea are having some types of hypersensitivity. So what they did, in order to treat all these things, they have isolated the toxins from the jellyfish, and they injected this isolated toxins to a dog. What happened, as soon as they injected this purified toxins from the jellyfish to dog, the dog immediately start reacting with the vomiting, diarrhea, and asphyxia, and sometimes they also let this also lead to the death of the dog. So all these symptoms caused in the dog were termed as anaphylaxis, which is actually just the opposite term or the analogous terms for the prophylaxis, which means that some or the hyper secretion or the hyperactivity of something. This finding, because of this finding, Paul Pater and Charles Richet were awarded with the Nobel Prize for the physiology or medicine in 1913. So this was a brief history about the hypersensitivity. There is not a single type of hypersensitivity, rather this hypersensitivity is of many types. So to deal with this, Gell and Combs has classified the hypersensitivity basically into four parts. Previously it was believed that hypersensitivity is very much restricted to the humoral immunity, but the last type of hypersensitivity, as per the Gell and Combs classification and also for the mechanism of their action, which is the type 4 hypersensitivity, is actually the cell mediated hypersensitivity, whereas the other basically include the immunoglobulin or antibodies, or specific to the B cells, or first activation starts with the B cells. So we all know that the immunological response where the B cells is having a major role is termed as the humoral immunity, whereas the one where the T cells are having a major role are known as cell mediated immunity. So we will be discussing in this video about all these four types of hypersensitivity one by one. So type 1 hypersensitivity is little bit complex, and there are so many things to know about the hypersensitivity in type 1. So we will discuss in detail with the type 1, and when moving toward the type 2 and type 3, yeah, we already have discussed somewhat about all these mechanism in my previous videos, like in complement system and how the recognition of the antibody antigens takes place. So I will mostly focus on the type 1 hypersensitivity, and at the end we will come back to the questions which we have seen in the previous slides. This table I have taken from the Cube, and I most favorite thing about this table is that I like this table very much, because once you will go through whole the process of hypersensitivity, this table is the complete explanation of the all types of hypersensitivity. Or if you want to revise before, just before the examination, this diagram, the diagram depicted here along with the explanation which is in the table, is complete, which is there in the further upcoming pages, or the upcoming pages is dealing about the hypersensitivity in Cube. So moving to the type 1 hypersensitivity, or this is also known as immunoglobulin E mediated hypersensitivity. So what happens whenever any allergens enter inside our body? Allergen is also a type of foreign particles, so it will also be recognized by the B cells. So this after recognition by the B cells, so we all know that the B cells will be having antibody on its surface. So what will happen when the allergens will come and bind on the antibodies? It will be taken inside by the process of phagocytosis, or simply they will be internalized through the process of antibodies. And after processing of this and allergens, this will be expressed on the surface of B cells through the help of MSC class 2 molecule. Now what happens when MSC class 2 molecules expresses something? It will invite the T cells, or the even the expression of any types of antigens by either MSC class 1 or class 2 molecule will invite the activation of the T cells. So this thing we already have discussed in antigen processing and presentation, so I'm not going to discuss more about this. And if you missed that video, you can go to that video from the I icon, which is just now popping on the top right corner of your screen. What happens after the activation of T cells? T cells start secreting cytokines, specifically interleukin 4. The function of interleukin 4 is that it will bind on the surface of the B cells, and it will induce the maturation of the B cells. So what will happen? The B cells will differentiate itself into memory cells and the plasma cells. So we all know that memory cells will be remain reserved inside our body when second interaction of the allergens will be there, or antigens will be there. It will be activated more frequently. Now after that, these plasma cells will start secreting the immunoglobulin. So this time the immunoglobulin will be immunoglobulin E. The specific types of immunoglobulin E will be secreted in response to the allergens. After the secretion of immunoglobulin E, when there will be more amount of immunoglobulin E, it will either go and bind on the Fc receptor present on the mast cells or the basophil cells. So there is a special configuration in the Fc region of the immunoglobulin E, which make it more favorable to bind with the Fc receptor, particularly for the immunoglobulin E, which we will discuss in a bit later. Now what will happen? Second time, when the same allergens enter inside our body, and we already have immunoglobulin E inside our body, so what will happen? This immunoglobulin E will be cross-linked by the presence of this allergens. After the cross-linking of antibodies E in presence of allergen, what will happen? Degranulation will take place. So we can see the grayish matter present inside this basophils or mast cells. This will start releasing outside. This histamines actually gives signals to the nearby cells or even the mast cells also to show the symptoms for the allergies. So what we have now concluded in the type 1 mandated hypersensitivity, that what happens when allergens bind over the immunoglobulin E, it will be taken inside the B cells. B cells will activate the T cells. T cells will then secrete out interleukin 4, which is a cytokine. Cytokines will induce the differentiation of B cells into memory cells and plasma cells. This plasma cells will be secreting the immunoglobulin E. This immunoglobulin E will then bind over the Fc receptor present on the mast cells, and after that, when again the allergen will enter inside our body, then this allergen will induce the cross-linking of the immunoglobulin E, and this will secrete the histamines, which will then lead to the stimulation of the smooth muscle cells, small blood vessels, mucus gland, because of which we start sneezing and all. Blood platelets will be activated, so that we start inflammation is also responsible for this. Nerve cells endings will be stimulated, which will secrete some of the ions like calcium ion, and also will stimulate the other symptoms inside our body related to the hypersensitivity. Another is the increased secretion of the eosinophil. So it is in layman language we say that a person is suffering from eosinophilia. So actually that is not eosinophilia, rather there is an increased secretion of the eosinophils, which is one of the marker to identify the allergic response. So this is the whole simple mechanism which happens in the type 1 hypersensitivity. Now, moving to the components, what are the components which are there inducing the type 1 hypersensitivity? So, there are mainly 4 components: allergens, reactogenic antibody or immunoglobulin E, mast cells and basophil cells, immunoglobulin E binding Fc receptors. So, we will be discussing about all these things one by one. So, first moving to the allergens. So, what are the allergens? Allergens are also termed as atopy or simply the excessive functioning of something or immunological functioning. So it may be also heredity, and this hereditary types of hypersensitivity is basically related with the immediate hypersensitivity or type 1 hypersensitivity, where there is inappropriate production of immunoglobulin E. So as this is hereditary, and so it means that it will be, or some genes will be responsible for the hypersensitivity. So when we will go up to the hereditary, then there comes a point that which types of gene is there. So on a deeper study it was analyzed that 2 chromosomes are actually involved in hypersensitivity. One is the chromosome 5 at the locus Q, and another is the chromosome 11 at the locus Q. Chromosome 5q is basically responsible for the secretion of the cytokines including interleukin 3, 4, 5, 9, 13, GM, CSF. So all these cytokines are involved in the hypersensitivity reactions, either in the maturation, as we have seen in the with the interleukin 4, which is involved in the differentiation of the B cells, and these B cells on differentiating into the plasma cells will secrete the immunoglobulin E, which is required for the hypersensitivity. Similarly, the chromosome number 11q are involved in the B chain formation, or the B chain synthesis is completely controlled by chromosome number 11q. So what is the role of beta chain in the hypersensitivity? We will discuss in the section of Fc receptor. Now when we are talking about the allergens, so there are multiple allergens. So most of you will be allergic to something. Some are allergic to some medicine, some are allergic to some food, paper, or black pepper, black pepper, or egg white. So these are some common allergens, and the most common I have noted only few of them, like ryegrass pollen, codfish, ragweed pollen. So but this is not limited up to here. There are lots of, or there are a long list of the allergens. So before without focusing on this particular type of allergens, I will focus on the mechanism how they activate. So what actually happens? Allergens either taken inside through inhalation, or taken in the form of the food also, basically. But there are also some surface, or the skin is also sensitive to some of the allergic responses, like some of the cosmetics also shows such types of reactions. So I will be basically focusing on such on the allergens which are taken inside our body. So what happens when they will be taken inside our body? So suppose the allergens is taken through the inhalation. So what happens? The hair present inside our nostrils will trap them, and the suppose this is the ragweed pollen. So pollen grains, we all know that is covered of two layers, exines and entines. So an outermost layer is a very hard covering. But what happens if this layer will be present, their hard covering will be there, then it will not show any allergic reaction. But as soon as these allergens are taken inside, it will be covered by the mucus present inside our nostrils and also the food pipe, or sorry, through the digestive tract. So what happens after being coated with the mucus? Mucus also consisting of various lytic enzymes, so it will lead to lysis of the outer wall of the pollen grains, and it will start secreting the toxic or the allergen product inside our body. So from here the allergic response shows. So basically, on the basis of the population being affected of these allergens, allergens were classified into two major classes. One is major allergens, which affect a majority of the population even when present in the minute concentration, whereas another is the minor allergens, which include RA3, RA4 and RA5, and it needs a high concentration to be affect any individuals, and this is very rare, or very rare population are being infected of this. So why EK fraction it is known as? So we will discuss it in just few after few more slides. Now next thing is that first component was what? First component was the allergens, which we have completed. Now second component for the hypersensitivity or type 1 hypersensitivity is the reasoning antibody, which was well explained by K. Prostnich and H. Kastner in 1921 with the help of skin well and skin flare reaction. So what are these two reactions? It is also advised when suppose you are having some new cosmetics, so it was advised that use some of the of that cosmetics in your skin area, and if you feel any rashes like thing or any itching, then it means that you are allergic to that cosmetic. So simple concept was there with the PK reaction. So because it was proposed by Prostnich and Kastner, so their first alphabet PK. So this reaction with well and flare reaction is popularly known as the PK reaction. So what happens in well? In well, there will be the swelling on the skin if we got the inflammation or small swelling on the skin, or if we have flare or the erythema or reddening or redness of the skin, then both of these together are termed as PK reaction. So how it was confirmed that this PK reaction or this allergic reaction was because of the immunoglobulin E. So to prove this on the basis of PK reaction, two scientists was there, K and T Ishizaka. In 1960, what they did, they have isolated serum from the atopic or from the atopic human or the human who are allergic to some allergens. So what will happen when a human will be infected or will be taken inside some of the allergens? Then what will happen? These allergens will lead to the production of, or they will start their own mechanism, and in response of this allergen, as we have seen in the previous slides, that there will be activation of the B cells, and these B cells will be secreting antibody, specifically immunoglobulin E. This immunoglobulin E, what will happen if I will design any antibody against this anti-immunoglobulin E, then this will stop the allergic reaction. So with this concept and with the help of PK reaction, what this Ishizaka has done, they have taken the serum from the atopic human. So the serum will be what? The serum will be consisting of all the 5 immunoglobulin E, M, G, D and E. After that, what he did, he was knowing that though that was not well proved on that time. So basically what will happen, along with this five antibodies in the atopic human serum, there will be excessive amount of immunoglobulin E as compared to other antibodies. So what he did, he has taken a rabbit and he has immunized a rabbit against the immunoglobulin E. So definitely, if a mouse is being inoculated with the immunoglobulin E from human, then definitely this and a rabbit being very because we know that for having an antigenic response or immunogenic response, there should be phylogenetic difference, or the organism must lie more the distance between the phylogenetic tree, more the serum will act as the allergic antigens for that particular organisms. So similarly here, what will happen when they have inoculated immunoglobulin E inside a mouse, then what will happen, this rabbit will be having antibody against immunoglobulin E. Now after that, what they did, when they have inoculated the anti-serum from the rabbit, this anti-serum will be having, anti-serum means what? Anti-serums means the serum or the anti-immunoglobulin E, or the antibody against this immunoglobulin E which was from the human serum. That's why I'm using the term anti-serum. So this anti-serum will be having affinity, or will treat this immunoglobulin E as an antigen. So what will happen when we will be inoculating this serum from the rabbit? So it will bind with against this this immunoglobulin E, and as this if there will be no free immunoglobulin E will be there, so this immunoglobulin will not be able to bind with the mast cells or the receptors present over the mast cells. So there will be no any allergic reaction. But when the same anti-serum is used against the rest of the four immunoglobulins, then even in that condition, will and flare reaction or the PK reaction was shown, or we can say the allergic reaction was shown. So all this experiment was compiled in a small table, where what we have seen, we have taken the serum from the atopic human and one from the non-atopic human. So if we are not treating with any, or there is no treatment of the atopic, then what will happen? In absence of allergen, simply there will be no PK reaction. But when again, when there is no treatment or no anti allergic or anti immunoglobulin E is there, then in presence of allergen, PK reaction will be there. If a human is non-atopic, then even in the presence of allergen, there will be no PK reaction. But in the in the fourth case, what will be happening? With the atopic person, if we have treated this atopic person with the rabbit anti-serum, then what will happen? This anti-serum will go and bind to the, or will affect the immunoglobulin E present in the human atopic serum. So this will, even in the presence of allergens, will not show the PK reaction. But at the same time, when a rabbit anti-serum will be used against immunoglobulin M, G, A and D, then in the presence of allergen, this PK reaction will be there. So this experiment by the Ishizaka has proved that for the allergic reaction, immunoglobulin E is particularly responsible. Now coming to the third component, which is the Fc receptor. Why only immunoglobulin E is specific to bind with the Fc receptor? So when we will take a close look with the immunoglobulin G and immunoglobulin E, what we will find here? We find here one extra constant region is present in immunoglobulin E, or the C4 region is there. This is particularly responsible for to be recognized as a marker for to bind over the Fc receptor on the mast cells or basophil cells. So moving toward the third component, which is the receptor, which is here actually two types of receptors are there. One is the high affinity immunoglobulin E receptor and another is the low affinity immunoglobulin E receptor. High affinity means what? Even the immunoglobulin E is in very low concentration, it can easily bind to the Fc receptor. Whereas if there will be very low concentration of the immunoglobulin A, concentration is low, then it will not able to bind with the Fc receptor. There is a requirement of high amount or the high secretion of immunoglobulin E. When we will come and look toward the structure of the immunoglobulin E, Fc receptor, then what we will see here, two chains are actually present here. One is the alpha and another is the beta chain. In alpha chain, you can see here immunoglobulin like domain is there, which is approximately of 90 amino acid chain, and this two domain which resembles like the immunoglobulin, because of this two domain, this receptor is placed under the immunoglobulin superfamily. And moving back to the slide, when what we have seen here one extra constant region. So this Fc receptor 1 or high affinity Fc receptor, these two domain will bind with the constant region 3 and constant region 4. This is how this will recognize the immunoglobulin E receptor even when the concentration of immunoglobulin E is very less. Now, what is the role of beta chain? Beta chain is actually responsible to connect the alpha chain with this gamma chain. So there are two types of or dimerization of the gamma chain is there. Both are connected with the help of disulfide bond. This gamma chain is very important. So what happens after the binding of immunoglobulin E with this receptor? This beta chain will pass the signals to gamma chain, which is the dimer, and the internal part or the cytoplasmic part of the gamma chain is having a domain, specific domain ITEM, which is the immunoreceptor tyrosine based activating motif. So this is what this will act as a signal molecules, and this will induce the liberation or the secretion or the degranulation, better I use the term degranulation of the histamines, which further shows the symptoms for the hypersensitivity. So this ITAM will be phosphorylated after the binding of the immunoglobulin E, and this will pass further signals to the nearby signal molecules, and this signal molecules will then induce the internalization of calcium ion. This calcium ion will further activate the other responses involved in the hypersensitivity. Moving to the other type of receptor, which is the low affinity receptor for the immunoglobulin E, will be required when there is very high concentration of immunoglobulin E, and this basically recognize the constant region 3 of the immunoglobulin E. So these are the two different types of receptors which are involved in the binding of immunoglobulin E over the surface of plasma cells as well as basophil cells. Now, last component, which is I am always using the term plasma cells and the basophil cells. So these plasma cells and basophil cells are basically involved in the hypersensitivity. How we can analyze this? This was analyzed by the help of autoradiography. So what it was done, when a isotopic or radioactive leveled immunoglobulin E were inserted inside the cell, then what it was analyzed that this is going and binding only with the plasma cells or the basophil. This was further later on confirmed by the structure or the morphology of the basophil cells, which are having which got dyed with only basic dyes, for what by which they got the name basophils. So likewise it was analyzed that these two cells, plasma cells and basophils, are the major requirement or one of the component of the type 1 hypersensitivity. Now finally, moving toward the mechanism of allergic reaction. What is the basic requirement of the allergic reactions? For any allergic reactions, once the allergens has entered inside our body and once the immunoglobulin E is been activated, the major requirement is the cross-linking of the immunoglobulins E or these receptors. So even in the absence of allergens, sometimes what happens, this hypersensitivity reaction can take place. How it can take place? So basic requirement is what? Cross linking. So this cross linking also takes place in presence of allergens. It can also take place in the presence of some a particular type of antibody, which is known as antibody mediated hypersensitivity, which we will discuss just later on. This antibody can bind to the Fc side of the antibody, or it can also bind to the Fab region of the antibodies, or there may be also that directly the receptors are cross linked, and or there will be chemical linking of this and of these receptors together. So all this mechanism of cross linking will lead to the degranulation. So here is a picture or a summary of what we have seen about the cross linking. So this cross linking can be an allergen, it can be anti-isotypic antibody, anti-idiotypic antibody, or any chemical reaction can be there, or directly the anti-receptor antibody can also activate the hypersensitivity, which will enhance the calcium ion influx by the ionophore, or simply taking in of the calcium ion through ion channel known as ionophore, which will increase the membrane permeability, and further responses will start occurring just after this reaction. So these were all about the type 1 hypersensitivity. Moving forward with the type 2 hypersensitivity. So let's start with the type 2 hypersensitivity, what exactly it is. This is a very common reaction which you all will be knowing about that blood transfusion reaction. So we all know that each cell has a certain marker. So just like that, RBC also has a glycoprotein over their surface, and O antigen is having this much of lipid or glycoprotein, which is common in A as well as B. So only difference lies with the A and B is the presence of an acetyl galactosamine in A antigen, and in B the antigen will be the galactose. So what will happen when this N-acetyl galactosamine will be diffused inside the person with B antigens? This N-acetyl galactosamine will act as the antigens. Similarly, when B antigens will be diffused inside the A or the patient having the A blood group, these galactose will act as the antigens. So this is the reason why O can be a universal donor, because it do not have any of this allergens, neither N-acetyl galactosamine nor the galactose. So this table I think you already will be knowing about this, so I'm not going to discuss much about this. So I'm directly focusing on the hypersensitivity. So here the hypersensitivity will be brought about by three mechanisms. One is by the activation of complement system, which we already have discussed in our previous classes. Another is the antibody dependent complement activation, and lastly the opsonization. So opsonization is also somewhere related with the complement system. So in the complement system, we already have discussed that a smaller component of the complement system like 3A, 4A and 5A are involved in the activation of some inflammatory response or the opsonization. Even sometimes what happens, some of the antibody goes and bind to the surface of the cells, and it will lead to the opsonization or invite the phagocytosis to be engulf these cells. So this is known as opsonization. So here the antibody dependent cytotoxicity also means the same. Here the killing of cell will be there, and this killing of the cell or this cell will be killed by binding of the antibody over the surface over the receptor of the cells, and instead of opsonization or instead of inviting the macrophages, what will happen? Directly the killing of the cell take place. So this is known as antibody dependent cell mediated cytotoxicity. So this mechanism all happens in the type 2 hypersensitivity. Finally, the third one, which is the third hypersensitivity, it is also a complement system induced one. So we already have discussed about the complement system, that each component of the complement system will be having two components. One is the bigger and one is the smaller one, with the exception of the 2A, where we have seen that A part of the complement system will always be smaller, but with the what is the exception with the complement system 2A will be the bigger one. So we already have known about all these things, and as in the just like that of the transfusion or the blood transfusion reaction, here also the smaller part of the complement system will go and activate the type 3 hypersensitivity. So what happens, suppose any allergens is been taken inside our skin, so this will be recognized by the antibody, and this antibody will activate the complement system. And after activation of the complement system, this 3b C3b will remain bonded with the complement system, whereas the smaller part of this, like 3a, 4a and 5a complement system, 3a complement system, 4a complement system, 5a will induce the degranulation of the mast cells. And once this degranulation will take place, histamine, which is a major component of this granules, it will go and bind over the smooth muscle cells, and it will also activate the inflammatory response by the secretion of neutrophils, because these antigens or this can also be allergens will invite the opsonization as it so we can say that type 3 is somewhat related to type 2 also. So here also we can see the complement system is playing a major role, and these neutrophils will go and engulf these antigens, and further the processing of immunological response will be there. So this was about the type 3 hypersensitivity. Last type of hypersensitivity, which is very different from the other, which is a delayed type hypersensitivity. So till type 4 hypersensitivity, what we have seen that humoral immunity was involved, but in type 4 hypersensitivity, cell mediated immunity will be there. So here a particular class of basically type the response will be by the T helper cells 1 subclass will be involved in the type 4 hypersensitivity. So this is why this is known as delayed type hypersensitivity, because what happens, I am just putting in one of the example of a poison from the auk. So this poison from the auk will be having the pentadeca catechol, which when enters inside the skin will induce the delayed type hypersensitivity. So what is the significance of this? Why, if there is a delayed type hypersensitivity, and when we are not able to recognize this hypersensitivity, because more rapidly the symptoms will be recognized, we will able to treat the hypersensitivity. But what will happen when there will be delayed type? So this is also significant when we will be invaded by the parasites or by the some bacteria which remain inside the cell, and inside the cell it is very difficult for the antibody to enter inside the cell to kill the bacteria inside the cells, until unless this bacteria will undergo proteolytic activity. As in the phagocytosis we have seen that the bacteria will be undergo with the help of lysosomes, it will be broken down into the small component, and it will be expressed on the surface of the cell with the help of MSC molecule. It is not possible to kill the bacteria. So sometimes this such types of parasitic bacteria when it will remain inside the cells, so what will happen, this delayed type hypersensitivity actually activate or will lead to the excessive secretion or the excessive stimulation of the macrophages. These macrophages will be induced by the cytokines released from the type 1 or helper cells, T helper cells. This macrophages will lead to the phagocytosis of the cells, and along with this macrophages will also release some of the the lytic enzyme. So the delayed type hypersensitivity, why it got delayed, why basically of 48 to 72 hours, because this is the time required to increase the concentration of the macrophages inside our body. This will keep on releasing a number of lytic enzymes, and when very high amount of lytic enzymes will be secreted, this will lead to the lysis of the cells, which will be invaded by the parasites. So finally the parasites will come out of the cells because of the lysis of the cell, and this parasite can be killed. So this is also a significance of type 4 hypersensitivity. So this is just an example where we can see that the entry of pentadecacatechol, which is toxin to a human, when it will come inside, it will release the toxins, and this will induce the activation of delayed type T cells. So there are also few cases where TC cells are also involved. So basically we can see that T cells are involved. So this is also known as cell mediated hypersensitivity. So this will lead to the release of number of cytokines, and these cytokines will invite or it will activate the macrophages, and this macrophages will go and will accumulate in toward the site where the toxins are present, and it will lead to the lysis of the cells, which is been internalized with the cytotoxins. So these were all about the hypersensitivity. So moving to the slide where we were having the questions, and I know that after this you will be able to answer all the questions by yourself. So you all go through all these questions, and you can comment the answer in my comment box. In my next class I will uh, start with the explanation of these questions, and then we will move toward the next topic. So hope so you like this video. So in next class we will be discussing about all these questions. You can also try to pause my video and can go and read out all these questions, and next class we will deal with this question. So don't forget to subscribe and share my channel. Till then, goodbye and be yourself.